Welcome back. In this session, we're going to be talking about the control of ventilation. And it's a remarkable fact that the gas exchange of the lung is so carefully regulated in spite of considerable differences in activity during the 24 hours, rest, exercise, sleep. In spite of that, the PCO2 of the arterial blood is probably kept to within 3 or 4 millimeters of mercury over the whole period. And this remarkable degree of regulation of gas exchange is brought about by the control of ventilation. And in this rather whimsical cartoon you can see at the bottom right there, that shows the way in which the blood gases are being scrutinized and ventilation is altered as a result. So let's look first of all at the system, the respiratory control system, which is shown here. And we can start with the sensors, bottom left. And these look at the gas exchange in the blood. We've got what are called chemoreceptors, and I'll go into those in some detail. They look at the levels of PO2, PCO2, pH in the blood. You've got receptors also in the lung, which look at the movement of the lung, and other receptors as well, for example, from exercising muscles. And these sensors send an input to the central controller in the brain. Now, the central controller, as we'll see, is mainly located in the brain stem, the medulla and the pons, but there are also influences from other parts of the brain. And that's where the information from the sensors is processed and the brain works out what the body really wants in terms of gas exchange. And so the output of the central controller goes to the effectors. The effectors are the muscles of respiration. Now, of course, we've dealt with them in some detail. We're not going to spend a lot of time on those now, but we'll just review briefly the respiratory muscles. Now, one of the important features of this respiratory control system is that it is a negative feedback system. What do I mean by that? Well, suppose the PCO2 in the blood starts to rise, perhaps because during sleep there's some obstruction to breathing. As a result of that, the sensors will send, the chemoreceptors will recognize the increase in PCO2 in the blood, and they'll send the information to the central controller, and the central controller will increase the ventilation by increasing the output to the respiratory muscles. The result of the increase in ventilation is to reduce the arterial PCO2, and in that way the system is returned to its normal situation. So that's an example of negative feedback. Now let's look at the central controller in more detail, and here first of all is a very simple diagram of the brain for those of us who don't see it all the time. The brain has a brain stem made up of the medulla, or the medulla oblongata, and the pons. The medulla is an extension of the spinal cord, as you can see here. And then there are other parts of the brain, the midbrain, the forebrain, and so on. But we're going to be mainly concerned with the medulla and the pons, although as we'll see, there are influences coming from the forebrain, the cortex, under some conditions. So let's look at the rhythm controllers in the brainstem. Now, I have to say that I find this a somewhat confusing area, partly because so much research work is going on all the time. People are still working out the details of the central pattern generator, the controlling system in the brain. But this is, I think, a reasonable way of looking at it, taking into account the more classical statements that have been made and some more recent work. And so in the medulla, we've got two groups of cells. They're sometimes called respiratory centers, although people don't particularly like that term. These are a collection of cells, and one is the dorsal respiratory group in the medulla, dorsal meaning towards the back in the upright man, the dorsal respiratory group, and these seem to be particularly associated with inspiration. Then we've also got a ventral respiratory group of neurons which seem to be particularly associated with expiration. Now, more recently, people have spent a good deal of time working on a thing called the pre-Botzinger complex. And this is thought to be very critical in terms of the central pattern generator. It's also located somewhat ventrally in the medulla, and researchers have carried out experiments on newborn animals where they've taken slices of the brain and shown that they can pick up areas in this pre-Botzinger complex which are undergoing intrinsic rhythm. In other words, they can put an electrode on this group of neurons and show that it is an intrinsic pattern generator, a little bit like the heart, which has a generator. So the pre-Botzinger complex now is being studied very extensively, and some people believe that that probably is where the intrinsic rhythm, the central pattern generator, is located. As we've learnt from previous sessions, during resting breathing, inspiration is active, but expiration is passive. During expiration, the lung returns to the normal equilibrium position for the lung and chest wall. And so if you look at the output of the phrenic nerve, for example, what you'll find is that the impulses coming from the brain gradually increase in frequency during inspiration in a sort of ramp type pattern, and then at the end of inspiration the impulses go away, and during expiration the system is quiescent. Of course, on exercise, when expiration has to be active, then expiratory impulses also occur. Now in the pons there are two groups of neurons, centers, that classically have been thought to be important, although exactly how important they are in normal breathing is still, in humans, is still not clear, because all these experiments necessarily are done on animals. And in the lower pons, there's a thing called the apneustic center, which is thought to have an excitatory function. If you section the brain of an animal above the apneustic center, you get a series of prolonged gasping inspirations. And occasionally patients with severe brain injury will show the same pattern. So under some conditions, the apneustic center may be important. And there's also a thing called the pneumotaxic center, higher in the pons, and it's thought that this can inhibit inspiration under some conditions, and possibly the pneumotaxic center is associated with the fine control of the frequency of breathing. But I should emphasize that I think most people who work in this area still don't fully understand the rhythm control in the human, because most of this work has been done on animals. Now, in addition to these controllers in the brainstem, other regions of the brain can affect respiration, and we should just mention these. First of all, the cortex is very important, and we're all familiar with that. We know that we can hyperventilate if we want to. In fact, it's not difficult to hyperventilate over a minute or two, and you can drop your PCO2 in the arterial blood to close to half the normal value. Hyperventilation is very effective in changing gas exchange. Hypoventilation, for example breath holding, is much more difficult. Although there are some of these divers who go down to great depths now, breath hold diving, and they can hold their breath for many minutes. So under some conditions, humans can control their hypoventilation as well as their hyperventilation. In addition to that, there is the limbic system and the hypothalamus, and these have to do with emotional states, and we're all familiar with the fact that someone who is very excited or is very afraid, something like that, they will change their ventilatory pattern, and that's because of input from these other systems. So that's all I need to say, I think, about the central pattern generator, the respiratory control system, and the central pattern generator in the brain. And now let's move to the effectors, which are the muscles of respiration. Now, as I said, we don't need to spend much time on these because we looked at these in detail in the session on ventilation. And just to recap though, the most important muscle of inspiration is the diaphragm, you recall. And one of the important things about the diaphragm is that it's supplied by phrenic nerves, which originate high in the cervical region of the spinal cord, so that damage to the spinal cord in the thoracic region, for example, spares the phrenic nerves, and therefore breathing can continue, and that's very important in the survival of people who develop this kind of injury. In addition, inspiration is assisted by the external intercostal muscles, you recall. Expiration is normally passive, as we've said, but during exercise, for example, when expiration becomes active, then the abdominal muscles become particularly important. They're the most important muscles of expiration. They're assisted by the internal intercostals. And we shouldn't forget also the accessory muscles of ventilation, muscles in the neck, for example, which support the upper ribs, the first rib particularly, but they come into play during vigorous exercise, for example, and also patients who are severely short of breath can be seen to be using the accessory muscles. So we don't need to say any more now about the effectors. Let's move on and talk about the sensors, and this is a much bigger topic. So the sensors can be divided into 4 main categories. First of all, there's the central chemoreceptor that I'm going to be talking about in detail. Then we've got 2 sets of peripheral chemoreceptors. We've got receptors coming from the lung, sensing the expansion of the lung, for example, and other receptors coming from muscles, limb movement, and baroreceptors, other sensors as well, which we'll mention briefly. So let's now move to the central chemoreceptor. And first of all, what do we mean by a chemoreceptor? Well, a chemoreceptor consists of specialized tissue that responds to a change in the chemical composition of the blood or some other fluid. And in fact, we'll see that as far as the central chemoreceptor is concerned, it's the extracellular fluid of the brain that's the important fluid. As far as the peripheral chemoreceptors are concerned, it's the arterial blood that is the fluid that is sensed. So we've got the central chemoreceptor and the 2 peripheral chemoreceptors. Let's look at the central chemoreceptor. Now here's a nice diagram showing the central chemoreceptor, which is embedded in brain tissue, as you can see. It's in the ventral part of the medulla. It's actually only a few millimeters below the surface. And if you take a pledget of cotton wool, which has some acid or CO2 solution on it, and place it on this part of the brain, this part of the medulla, then you can see in animal preparations a rapid increase in ventilation occurs within a few seconds. So there's a central chemoreceptor embedded in the brain tissue here, and this is a very important receptor. In fact, this is the central chemoreceptor is responsible for most of the minute-by-minute control of ventilation over the 24 hours. The central chemoreceptor responds to pH in the extracellular fluid, which essentially is the pH of the cerebral spinal fluid here, CSF, meaning cerebral spinal fluid. We can't get at the extracellular fluid easily, but the cerebral spinal fluid is easily looked at. We can put a needle into the space and take some of it out if we wish. And it's the pH of the CSF which apparently has a large effect on the response of the chemoreceptor. Chemoreceptor also depends to some extent on the blood flow through the brain perhaps, but mainly and metabolism, local metabolism perhaps, but it's mainly the pH of the CSF that's important. And the way it works is that you have blood in one of the smaller blood vessels in the brain. It turns out that the blood vessels in the brain have a remarkable barrier called the blood-gas barrier, which limits the permeability of many substances across the wall of these blood vessels. The cerebral blood vessels are special in that regard. And for example, they don't allow hydrogen ions or bicarbonate ions to permeate this blood-gas barrier. On the other hand, molecular CO2 can easily diffuse out from this central region, from the lumen of the blood vessel, into the extracellular fluid and into the CSF, and an increase in PCO2, of course, will reduce the pH, will increase the hydrogen ion concentration, and will reduce the pH of the CSF. So the sequence is that if the PCO2 in the arterial blood increases, more CO2 diffuses out into the CSF, the pH is decreased, and this stimulates the chemoreceptor. Now, the pH of the CSF is normally about 7.32. It's rather lower than that of blood, which of course has a pH normally of about 7.4. So the pH of the CSF is lower, and also the buffering ability of the CSF is much less than that in the blood. The CSF contains a lower protein concentrations, so a given change in PCO2 will cause a larger change in pH. Other things in the blood, the other blood gases, do not affect the central chemoreceptor. For example, the PO2, as far as we know, does not affect the central chemoreceptor, and pH, unless it's possibly moved over very large changes, also does not affect the pH of the CSF, and therefore does not affect the function of the chemoreceptor. So the chemoreceptor basically is governed by the CO2 level in the blood via its effect on the pH. An interesting thing happens if somebody is hyperventilating or hypoventilating for a long period. This sometimes occurs and therefore the CO2 rises. The CO2 sometimes rises in patients with chronic lung disease. But under those conditions, if the CO2 rises over a long period of time, and of course that causes initially a reduction in pH, the pH can be restored to near normal in the CSF by an organ called the choroid plexus. Think of the choroid plexus of the CSF like the kidney of the blood. The choroid plexus regulates the composition of the CSF. And so if the pH is moved, if for example if the CO2 increases over a period because of chronic lung disease over a long period, pH initially falls, but the CSF bicarbonate then can be reduced, and that can bring the pH back to near normal. And so what you often find in these patients is that they may have a chronically raised PCO2 in the arterial blood, sometimes known as respiratory failure, and yet the pH of the CSF is near normal, and therefore their ventilation is not increased nearly as much as you would expect from the level of the PCO2. So let's summarize some of the statements we've made about the central chemoreceptor. As I've said it, and this is here mainly if you want to come back and just review this, it responds to the pH of the extracellular fluid, which is broadly speaking that of the cerebrospinal fluid. The mechanism is that the CO2 diffuses across the blood-brain barrier, thus changing the pH of the ECS or the cerebrospinal fluid. The normal pH of the cerebrospinal fluid is about 7.32. It has little buffering power because of its low protein concentration, and the bicarbonate is controlled by the choroid plexus and can be altered if the pH of the CSF is changed over a long period of time. So that's the central chemoreceptor, very, very important factor in the respiratory control system. Now let's look at the peripheral chemoreceptors, and there are two sites of the peripheral chemoreceptors. The first is the carotid body, which is at the junction, the bifurcation of the carotid, the common carotid artery divides into internal and external carotid arteries, and that's where the chemoreceptors are located. They're very tiny, by the way. They're very, very small. That's where they occur. The others are the aortic body chemoreceptors near the aortic arch, as you can see here. These are not so important in humans. The more important of the two groups is the carotid body chemoreceptors. And you'll also see that and incidentally, the information from the carotid body chemoreceptors goes to the brain via the glossopharyngeal nerve, that's nerve number 9, whereas the information from the aortic body chemoreceptors is inputted to the brain via the vagus, that's cranial nerve number 10. You'll also see that close to the chemoreceptors are baroreceptors. We're not going to say much about those. They're much more important in cardiovascular physiology. Hear a lot about them there. But it is true that baroreception does affect the chemoreceptors as well. They meet, the outputs meet in the brain, and we'll say a little bit more about that in a moment. So let's look at the carotid body chemoreceptor in more detail. Here's a diagram of the carotid body chemoreceptor. You can see there are two types of cells. These are called glomus cells, and the type 1 glomus cells show a strong fluorescent reaction because of the presence of high concentrations of dopamine in them. And there are also type 2 glomus cells as well. Now, we're not sure about the exact mechanism of chemoreception, the response to the PO2, which is the important thing for these peripheral chemoreceptors, but it's generally agreed that one or two of these cells respond to the PO2, and the information is then sent to the central nervous system, the CNS here, and that's done, as we said, through a branch of the glossopharyngeal nerve. The carotid body has a very high blood flow, one of the highest blood flows in relation to its mass of any organ in the body. Having said that, the mass is very small, but the blood flow in terms of the mass is very high, and so the carotid body chemoreceptor responds essentially to the arterial PO2. It doesn't matter too much what the venous PO2 is, it's the arterial PO2 because the blood flow is so high, plenty of oxygen, and so it's the arterial PO2 that governs the response of the carotid body chemoreceptor. That's important in some conditions. For example, in carbon monoxide poisoning, where the blood may have a lot of carbon monoxide on board, and therefore the oxygen concentration of the arterial blood is relatively low. Nevertheless, the PO2 of the arterial blood is normal, and therefore the carotid body chemoreceptor is not particularly stimulated because it's looking at the arterial PO2, which is normal, and therefore breathing is typically not increased in carbon monoxide poisoning. Now, the output of the carotid body chemoreceptor, the response that goes up to the brain here, is plotted against the arterial PO2 here, and you can see it's very nonlinear, although it is interesting that there is some response of the carotid body chemoreceptor even at very high PO2s, and that's quite special amongst tissues of the body. But by and large, the response under normoxic conditions is very, very small, and therefore the carotid body chemoreceptor is not important in the normal control of ventilation when we're breathing air at at sea level. We're under normoxic conditions, and the response of the receptor is not important there. But as you reduce the arterial PO2, for example, by going to high altitude, then the response increases rapidly, as you can see. So it's a very nonlinear response. So again, just to recap on that, first of all, I didn't say, and I shall say it now, that in addition to the PO2, which is very important for the carotid body also responds to PCO2 and pH. Its response to PCO2 is less than that of the central chemoreceptors, perhaps 1/5 as much response of ventilation to a change in PCO2, an increase in PCO2, for example. Only about 1/5 of the increase in ventilation comes from the peripheral chemoreceptors, particularly the carotid body, although the response is rapid. And so if you've got a rapid change in PCO2, then the carotid body is important. And in fact, there's some evidence that even during the breathing cycle, the change in the carotid body response, because of the very small alterations in alveolar and therefore arterial PCO2 with the breathing cycle, apparently the carotid body can respond to those. Little response, and it also responds to pH as well, and that's important. I mentioned that the central chemoreceptor does not respond to the pH of the arterial blood, at least under most conditions, but the carotid body does, and that is important, for example, in metabolic acidosis. The increase in ventilation is caused by the action of the carotid body. Little response in normoxia, as I said. I emphasize the very high blood flow in relation to the mass of the organ. It therefore responds to arterial, not venous PO2, and it's got a fast response. So those are some of the features of the carotid body. Now, in addition to the chemoreceptors, ventilation is affected by lung receptors. And there are several of these, and they're listed here. First of all, there are the pulmonary stretch receptors. Now, the ends of the receptors themselves are probably in the wall of the airways, and they respond to the increase in volume of the lung. Some people call these slowly adapting pulmonary stretch receptors. Sometimes you'll see that term rather than stretch receptors. I personally happen to prefer stretch receptor, but a lot of people call them slowly adapting, and that means that if you change the signal on the receptor, if you change for example, the volume of the lung, the receptor will maintain responding at that increased level. That's a slowly adapting one, whereas a rapidly adapting receptor will initially increase its output and then it'll return towards normal. Now there's a classical reflex that should be mentioned. That's called the Herring-Brewer reflex. And the Herring-Brewer, or Breuer reflex, has to do with the fact that if you inflate the lung, then you inhibit further movement of the lung, further inflation. The animal will stop breathing sometimes. And that can be demonstrated, particularly in the rabbit, using the rabbit as an experimental animal, because there's a little slip of the diaphragm that you can measure the tension in, and that's the Herring-Breuer reflex. You can see it's also an example of negative feedback, because when you expand the lung, the reflex tends to prevent further expansion and further ventilation. And it was a very classical reflex. It was described way back in, I think, the 1880s. I can't remember exactly when, by Breuer. Breuer was the graduate student who actually did the work. Herring was his boss, so his name is attached to it. But that was one of the first reflexes demonstrated in physiology, actually, and particularly in respiratory physiology. And it was recognized that it had a negative feedback function. In addition to that, there are these irritant receptors, which are sometimes called rapidly adapting stretch receptors. Now, that name is given because although these mainly respond to irritants such as inhaled cigarette smoke, smog, toxic gases, cold air, these all stimulate the irritant receptors which are in the bronchial walls. Some people believe that they also respond to lung stretch as well, and that's why they're given this name. The result of these is often to cause bronchoconstriction. And in fact, some people think that the irritant receptors play an important part in asthma, which is a disease causing severe constriction of the airways. But you'll find, for example, in asthma, patients with asthma often will develop an attack if they breathe cold air, for example. That probably the irritant receptors are important there. There are also J receptors, or juxtacapillary receptors, as you can see here. The J receptors, the endings of the J receptors are in the alveolar wall, very close to the capillaries. The evidence for that is that if you inject a substance into the pulmonary circulation, you get a very rapid response of these J receptors. And the J receptors apparently cause rapid shallow breathing. And these may be important in some pathological conditions. For example, in pulmonary edema, where you get engorgement, a lot of fluid in the region around the pulmonary capillaries, the fluid leaks out of the capillaries into the interstitial space, it can presumably stimulate these juxtacapillary receptors and causes rapid shallow breathing. And rapid shallow breathing is very much a feature of pulmonary edema. People with pulmonary edema become very short of breath, very dyspneic, as it's called, short of breath. Also, this may be true in patients with interstitial lung disease, where the collagen is laid down in the alveolar wall, and this may well, we're not certain, but we think probably this stimulates the J receptors, and those patients typically have very rapid shallow breathing. So that may be one of the ways in which the J receptors work. There are also the bronchial C fibers. These are apparently similar to the J receptors, except that they are supplied by the bronchial circulation. So those are the lung receptors. But there are other receptors as well. There are receptors in the nose and the upper airway. Some of these are very like the irritant receptors. And for example, if you breathe very cold air, the upper airway may cause bronchoconstriction. If you breathe a noxious gas, a small amount of chlorine, for example, you may get constriction of the airways of the lung. There are also receptors from the joints and the muscles of the body, and these may be important in the increase in ventilation that occurs with exercise. There's a thing called the gamma system, which has to do with sensing the elongation of muscles. And some people believe that the gamma system may be important in the sensation of dyspnea. Dyspnea means the sensation of shortness of breath, difficulty with breathing, and some people think that the gamma system may be important there, although that's not certain. I mentioned the baroreceptors, and we saw that the baroreceptors are close, anatomically close to the chemoreceptors, the peripheral chemoreceptors. And the baroreceptors do play a role to some extent in the control of ventilation. For example, we showed in some experiments done on astronauts in space, and I'll mention those in a later session very briefly, we showed that the ventilatory response to PO2 is reduced in space compared with people standing at sea level. And that's because the baroreceptors in space are sensing a higher pressure in the blood vessels. That's because of the return of because the blood volume of the upper part of the body is increased in space because there's no dependent pooling of blood, and that's an interesting little feature that the response to PO2, which normally of course is done by the peripheral chemoreceptors that was modified in space. And also there are receptors that pick up changes in pain and temperature, and we're all familiar with that. A sudden pain will cause a gasp and perhaps a period of breath holding, for example. So these receptors are also important. Okay, now I've dealt with the main components of the respiratory control system. We looked at the, first of all, we looked at the central controller in some detail in the brain. We then looked at the effectors, the muscles of respiration, and we looked at the sensors, particularly the chemoreceptors, but other sensors as well. And now we're going to turn to the integrated responses, and here is the ventilatory response to carbon dioxide. So in this plot we have ventilation on this axis with the PCO2 here, and as you can see, as you increase the alveolar PCO2, so the ventilation increases. And incidentally, in experiments like this, where the lung is normal, we normally use the alveolar value as a measure of the arterial value. And you can also see that the slope of the line depends on the alveolar PO2. For example, if we drop the alveolar PO2 to around about 37 millimeters of mercury, there's a very steep increase in the rate of ventilation with a change in PCO2, much more so than when the PO2 is the normal value of 100 or even increased to 169 here. So there's interaction between the two stimuli here, the PO2 and the PCO2. Of course, the ventilatory response to CO2 is mainly the central chemoreceptors. The peripheral chemoreceptors make some additional response. The PO2 is only sensed by the peripheral chemoreceptors. Now, this ventilatory response to CO2 is very important. In fact, it is the level of CO2 in the blood that normally controls ventilation over the 24 hours of activity. It's the CO2 that's the most important variable. And we can summarize some of the features here. As I say, it's the primary factor in the control of ventilation. How do we measure it? Well, we can measure it by having a subject rebreathe from a bag. In other words, it's connected to a rubber bag which contains some CO2, and as he inhales the CO2, his ventilation increases, and then as he adds CO2 to the bag, because he's producing it from his tissues, so the PCO2 rises, and we can look at the response, the ventilatory response to the increase in PCO2. So that's a way of measuring the ventilatory response to carbon dioxide. We can also measure this, and I won't go into detail, using a technique where you suddenly occlude the airway during an inspiration. Now, you only do that for a very short period of time, but the inspiratory effort at the point of occlusion is measured, the pressure, and that pressure gives you a measure of the response to CO2. So that's another way of measuring the response to CO2. The CO2 response is altered by a number of factors. I've put a couple of them here. In sleep, for example, the response to CO2 is reduced, so typically the PCO2 will rise by 1 or 2 millimeters or 3 millimeters of mercury during sleep. The response to CO2 is reduced. Increasing age tends to reduce the ventilatory response to CO2, and there are genetic factors as well. In fact, there's a big difference, individual difference, between people in their response to CO2. In fact, if we go back to this previous slide, what we're seeing here is a mean of a number of measurements, but there's a good deal of variation between people, and that may be important in some kinds of lung disease. The CO2 response also may be affected by training. We do know that, for example, swimmers tend to have a reduced ventilatory response to carbon dioxide, and that may be part of the effects of training. It's also reduced by increasing the work of breathing. For example, if you ask someone to breathe through a narrow tube and you therefore cause respiratory obstruction, airflow obstruction, and the work of breathing is increased, those people will show a reduced ventilatory response to CO2, and that may be important in lung disease. Okay, so that's the response to CO2. Let's turn to the ventilatory response to PO2. Now here we've got measurements of ventilation here, ventilation measured against the PO2, and as you can see, as the alveolar PO2 falls, the ventilation increases. Now with a normal alveolar PO2, 36 or so, normally about 40, there's almost no response until we get to an alveolar PO2 of 60 or so, and it starts to rise. And that reinforces what I said earlier, that the normal level of ventilation under normal noxic conditions is not affected by the PO2 or the response of the chemoreceptors. But it's interesting that if you raise the PCO2, then the response to PO2 occurs at lower values. Here we've only raised the PCO2 to 43.7 millimeters of mercury, but now you can see the response occurs at a relatively high level of PO2, and this is even more marked when the PCO2 is raised to 48.7. So again, we can see an interaction between the two stimuli, the PCO2 on the one hand and the PO2 on the other. The response to reduced PO2, as I said, no role under normoxic conditions. It can also be measured by rebreathing from a bag. You have a bag containing a low oxygen mixture, and as you continue to rebreathe from it, the PO2 falls because you're using up some of the oxygen, and you can measure the response, the ventilatory response to the PO2 or the arterial oxygen saturation, which is often done. I've mentioned that its response is increased if the PCO2 is raised. There is an interaction between the two groups of stimuli. It's particularly important at high altitude, and we'll see that in a later session. When you go to high altitude, the ventilation increases a great deal, as a matter of fact, at extreme altitude, and this is brought about by the peripheral chemoreceptors. In fact, some years ago, some patients were treated for, I think it was asthma, they were treated by removing the peripheral chemoreceptors, or the carotid body receptors. It was not a good idea, but it was done, and it was shown that these patients had no ventilatory response to a low PO2. And that was a nice proof in humans that the ventilatory response to PO2 mainly comes from the carotid body, and certainly all of it comes from the chemoreceptors. The response to a reduced PO2 is important in some patients with chronic lung disease. I'm not going to go into this in detail, but some patients with very severe lung disease have an increased PCO2 and a very low PO2. And in some of those patients, a lot of the drive to breathe is coming from the low arterial PO2. Not so, of course, under normoxing conditions, as I've emphasized, but in these patients with a chronically reduced arterial PO2, some of the derived ventilation comes from the low arterial PO2. That's important because if these patients are admitted to or come into the emergency room and are given oxygen to breathe, which sounds like a very reasonable thing to do because their arterial PO2 is very low, they may stop breathing, or they may reduce their breathing to a very large extent. And this is well recognized. So what you need to do in these patients is monitor the PO2, the arterial PO2 and the arterial PCO2 carefully, and you normally give a relatively small increase in oxygen. You don't give them 100% oxygen to breathe, you give them 24, perhaps 28% oxygen, and monitor their blood gases. So those are the responses to PO2. What about the responses to pH? Well, these are not so important under normal conditions, but a reduced pH is sensed by the peripheral chemoreceptors, as I mentioned, probably particularly the carotid chemoreceptors. Of course, when the PCO2 rises, there is normally a fall in pH, and this is part of the stimulus to the peripheral chemoreceptor, and actually often it's quite difficult to sort out whether it is the PCO2 or the reduced pH which is responsible for the increased ventilation. But that's well seen in patients with metabolic acidosis that you'll remember we talked about when we reviewed the acid-base status of the body. In metabolic acidosis, the pH of the blood is low, there's acidemia, and often the PCO2 is low. So in those patients, the increase in ventilation clearly is coming from the reduction in pH. It's also believed, I mentioned that pH works through the peripheral chemoreceptors, but there's some evidence that if the arterial pH is sufficiently reduced to a very low level, the central chemoreceptors may be stimulated, and maybe some of the change in pH leaks across the blood-brain barrier, which, as I said, normally that doesn't happen. Let's turn now to the response to exercise, and this is a somewhat unsatisfactory area because I'm embarrassed to tell you that, at least in my opinion, we still don't fully understand the ventilatory response to exercise. Now that's a very remarkable statement, isn't it? Since all of us exercise and all of us are very much aware of the increase in ventilation when we exercise, but the reasons for the increase in ventilation are still not fully understood. First of all, the blood gases are normal during exercise, or at least in moderate exercise. When you go up to very severe exercise, blood gases may change a bit, but in moderate exercise, the blood gases are normal. The PCO2 is normal, the pH is normal, the PCO2, the PO2, and the pH are all normal in moderate exercise. The pH is normal except at very heavy exercise. Now, at very heavy exercise, you can get lactic acid production, and this will reduce the pH of the blood. And as a matter of fact, and we'll be talking about exercise a little bit in a later session, in very heavy exercise, the ventilation increases much more than you might expect. And that is because of the lactate. But that does not occur in moderate exercise. There's a small amount of lactate, but it's very small in moderate exercise. So what is responsible for the ventilatory response to exercise? Well, some people have suggested that it may be impulses coming from the cortex. We're aware of the fact we're exercising and we increase our ventilation correspondingly. And it's interesting that if you have a dog as a pet and you say to the dog, let's go walkies, the dog immediately starts ventilating, okay? He hasn't done anything yet. He just heard the term walkies and is aware of the fact that he's going to have some exercise, so he starts to ventilate. So clearly the cortex is important. Some people think that impulses from limbs are important in exercise. There's an increase in temperature of the body on exercise. That may be a factor. It's possible that there's a resetting of the CO2 reference level. In other words, we normally have the CO2 reference level at 40. Maybe it's reset on exercise, and that's the reason why ventilation increases even though the PCO2 remains constant according to our normal thinking. So the answer to all this is we don't fully understand what the response of ventilation to exercise is caused by, and maybe one of you out there will get a Nobel Prize one of these days from sorting that out. Now I'm going to finish by talking briefly about abnormal ventilation. I'm not going to go into this in detail, but I do want to say a little bit about sleep apnea because it's a very important topic and it occurs in essentially normal people. You may say, well, you're talking pathophysiology. Well, we're not meant, of course, to go into periods of apnea when we sleep, but relatively normal people do. And the commonest cause of, and it's extremely common. It's interesting that it's only been recognized in the last 15 or 20 years how common it is, but it's now recognized that it's extremely common, and you'll find sleep clinics all over the country helping people who develop sleep apnea. The commonest cause of sleep apnea is obstruction, and it's obstruction caused by tissue in the pharynx, which falls in during sleep and partially obstructs the airway. And snoring, of course, is a very common feature of this obstruction. It's often associated with obesity, although not always. Certainly it was initially described in obese people, but now people who are not obese frequently suffer from sleep apnea. It's important because it may result in sleep deprivation, which causes, as it says here, daytime somnolence, so that you get people who are sleepy. And obviously that's a serious problem if you're a truck driver, for example, and you tend to drop off during the day. That's a very serious problem. And in fact, some people have changes, and they've been measured, changes of cognitive function as a result of sleep deprivation. And also there are changes in behavior patterns. Some people they change their emotional states because of sleep deprivation. So it's an important area, perhaps not completely normal physiology, but it certainly occurs very frequently in people who otherwise appear to be normal, so it's something we should mention. There's also a form of sleep apnea called central sleep apnea. That's caused by respiratory depression during sleep. Do you remember I mentioned that the PCO2 tends to rise a little bit during sleep, and some people get a more pronounced depression of respiration, and that's called central apnea. You can distinguish between these two types, because if you make measurements of electromyograms, something like that, of the respiratory muscles, you can see that during obstructive sleep apnea, the subject is trying to breathe. The respiratory muscles are working hard against the obstruction. But in central sleep apnea, that's not the case. The respiratory muscles are not trying to overcome the obstruction. There is no obstruction, and it's in central origin. And finally, let me mention a type of periodic breathing which we see very commonly at high altitude in completely normal people. This periodic breathing is shown in the typical tracing here. Here we're measuring the excursions of the chest during breathing. Inspiration is down, expiration is up, and you can see that there are a few large excursions. This is done actually by having a tape around the chest. And then after a series of big breaths, there's a period of apnea, which may last 10, 15, even 20 seconds. And if you happen to be at high altitude and you're sharing a tent with somebody who shows sleep apnea, it's rather alarming, because he, after a few breaths, he stops breathing, and you wonder, is he ever going to start again? You know? But after 10 or 15 seconds, then these enormous big breaths occur. So this is called periodic breathing. It's almost universal at high altitude in normal subjects. You can see that in this particular tracing, someone was measuring the arterial oxygen saturation using an oximeter. That's a device that measures the color of the blood, and you can put a pulse oximeter on your finger, and you can see that the saturation is fluctuating the same rhythm as the periodic breathing, as you would expect, so that after the apneic periods, the saturation falls. Around about this point here, this particular subject was given oxygen to breathe. Saturation increased, as you can see, and the periodic breathing largely went away. And it's known that periodic breathing is certainly very greatly reduced, or perhaps abolished, by breathing 100% oxygen. So let me now just briefly summarize some of the points we've made on this, the control of ventilation. The first point we made was that it's remarkable how efficient the control systems of the body are, in that the arterial PCO2, for example, only varies by 2, 3, 4 millimeters over the whole of the 24 hours. So it's a very remarkable system, and this is brought about by having sensors which look at the level of the blood gases and other things in the body, state of the lung, and they send information to the control system. There are three elements of the control system, as I mentioned. There's the central controller, mainly in the brain, And there are the effectors, which are the respiratory muscles, which we now know all about. And there are the sensors, mainly the chemoreceptors, but other sensors from the other parts of the body, the muscles, for example, and joints and so on. We pointed out that the cortex has an important, or first of all I should say that the structures, the neurons responsible for the respiratory rhythm, the normal rhythm, are in the brain stem, in the medulla and the pons, particularly in the medulla. And there's a group of neurons called the pre-Botzinger complex, which is now believed to have a critical role in maintaining the normal respiratory rhythm. But the brain stem centers are also affected by other regions of the brain. For example, we pointed out that there's voluntary control of ventilation. It's very easy to hyperventilate over a period of 2 or 3 minutes if you want to. Not so easy to breath hold, but some people can do it for several minutes. We talked about the effectors, which are the respiratory muscles, and one point I didn't make, and I meant to, was that it's important that the respiratory muscles work in a coordinated fashion. And I say that because there's some evidence that newborn babies sometimes lack coordination in the action of their respiratory muscles. And this may be, some people think this may be, play a role in the infant respiratory distress syndrome, cot deaths, where an infant is found dead in the cot, tragic situation, and not fully understood. And it may be that these infants lack the coordination of the respiratory muscles which they should have. We then went on to talk about the sensors. We talked about the central chemoreceptor, very important, the fact that it responds to the pH of the extracellular fluid, essentially the same as the cerebrospinal fluid, and that the pH of the CSF alters considerably because the buffering of the fluid is low. We also pointed out that the choroid plexus, which determines the composition of the CSF, is able to change the level of bicarbonate, and that is rather similar, and perhaps I didn't say this, rather similar to the metabolic compensation of respiratory acidosis and alkalosis. This is compensation in the CSF by the choroid plexus altering the level of bicarbonate. We then talked about the peripheral chemoreceptors, the type 1, type 2 glomus cells, the fact that the peripheral chemoreceptors are the most important receptors, or the receptors that respond to the PO2 in the arterial blood. And for example, if you go to high altitude, it's the peripheral chemoreceptors that are responsible for increasing ventilation, but they also respond to PCO2 and to pH. We pointed out that the peripheral chemoreceptors have a very high blood flow and therefore respond to arterial PO2, an important point because in some dangerous situations such as carbon monoxide poisoning, the ventilation does not increase. You'd think it would. I mean, it certainly would be very helpful if it did, but it doesn't increase, and the reason is that the arterial PO2 is normal and therefore they don't change. We talked a bit about the pulmonary receptors, particularly the stretch receptors and the old classical Herring-Breuer inflation reflex. We then went on to look at other receptors from the body, from the joints and some pain and other receptors. We talked about the integrated responses. First, the response to an increased PCO2 and how this is affected by the PO2 level in the blood. We talked about the integrated response to a fall in PO2 and the integrated response to a fall in pH. We pointed out that much to our embarrassment, we don't fully understand, or perhaps I should say I don't fully understand, the ventilatory response to exercise, and we finished by talking about abnormal patterns of ventilation in obstructive sleep apnea, very common condition. Some people regard it as an epidemic, actually. It's so common. And we referred to the periodic breathing at high altitude. So that concludes our session on the control of ventilation, and I hope to see you next time.